introduce them. Thanks for letting me know. I have the pleasure to invite an old friend for our seminar talk today. Imke Schmitt is a professor at the Goethe University in Frankfurt, and also a researcher at the Senckenberg Research Institution and Museum, and is deputy director of the Biodiversity and Climate Institute at Senckenberg. She has been working on lichens for quite some time, finished her PhD at the University of Essen in 2002 on a project on evolutionary biology of an order called Pertusarialis. Then she moved to the US and worked for 3 years here at the Field Museum as a postdoc, with at that time a young curator starting here, and moved over to be a postdoc at an assistant professor at the University of Minnesota from 2007 to 2010. And since 2010, she is in Frankfurt in this dual role as a professor at the university and senior scientist at the Senckenberg Museum. So today she will talk about elevation is a driver of differentiation in the lichen symbiosis. Please, Imke.
Thanks a lot, Thorsten. So I am extremely happy to be back here at the Field Museum. I've already seen quite a few people I've known from 20 years ago, and I've gotten a lot of hugs, and it's just very wonderful to be here. This is the third time I had the honor of being allowed to speak here in the Field Museum seminar series. And after all this time, I am still interested in lichens and also still working on them. So today, I'm going to summarize some research we have been carrying on over the last maybe 10 years, where we have been interested in how a lichen holobiont can achieve climatic niche specialization. And for this, we have been working along elevation gradients quite a bit. So elevation gradients are used extensively in ecological research because they are great to study how different species and communities respond to climatic factors. And the life zones that we see along elevation gradients are hugely compressed here, as opposed to them being spread out across thousands of kilometers along latitude. So the elevation gradients are like natural laboratories for us to study how climate affects species and species communities. And this is especially useful if we have species that we cannot just take home to the lab and cultivate and expose to controlled conditions, such as lichens. So change along elevation gradients can take different forms. Abiotic change along such gradients is very often linear. For example, if you think about this most important fact that affects organisms' temperature. Whereas if we look at biotic changes along gradients, we very often see a non-linear relationship where we have like a stable state at one elevation, then we have a steep transition, and we see another stable state. And this is why we see, for example, elevational belts of vegetation along in vascular plant communities. So it can be quite interesting to compare what's happening and contrast what's happening here between what's happening here. And we can compare entire communities and find out what changes, or we can take individual species and see, for example, how their genomic composition changes as we go up an elevation gradient. We can also focus on these transition zones and try to understand which are the environmental conditions that bring about these biotic changes. So we are interested in this question: how do communities and individual species respond to elevation gradients? And the system we use is the lichen symbiosis. And I'm sorry for some repetition here. In my talk today, I will talk about distribution patterns within the lichen holobiont along elevation. I will talk about some physiological responses that we have seen along elevation, and about genomic differences between high elevation and low elevation populations. So the lichen symbiosis consists of the main primary fungal partner, which is giving the shape, or which is causing the shape of the lichen, which is called the microbiont, and a primary photosynthetic partner, which is included into the lichen thallus. This is very often the green alga Tribouxia, which I indicated here. And in addition to these primary bionts, we also have more or less specific additional fungi that can be present, and more or less specific bacterial communities, which is why it's probably better to always talk about the lichen holobiont, or people have even termed the lichen symbiosis a micro-ecosystem. And lichens are highly diverse in morphology and also in chemistry, as you can see, can you tell by these different colors and shapes of lichens you see here. They can colonize basically every terrestrial surface, from natural surfaces such as rocks and bark and leaves, and also unnatural surfaces if you just leave them around for a long time. They can also be found in virtually every ecological niche in the terrestrial world, which ranges from the wet tropics to deserts, Arctic and Arctic regions. And so it's particularly interesting to understand, how do they do this? How do they cope with all of these climatic niches? The distribution of some species can be really broad, and this is just one example of a taxon we have been working with for quite a while now. Umbilicaria pustulata, for example, is distributed all across Europe, from Norway to Italy and from Portugal to Ukraine. So you can imagine the diversity of climatic conditions that this species encounters. And in contrast to most vascular plant species, lichens also go across biome zones, right? You can find them in Mediterranean climate as well as in cold temperate, for example, same species. And we can often study the same taxon along latitudinal as well as altitudinal gradients. So here we wanted to know how the different components of the lichen respond to an elevation gradient. And we looked at fungal genomic changes, we looked at switches of the Tribouxioid photobionts, and we looked at bacterial community change. We had a gradient that covered multiple biomes, and we wanted to know, are these changes that we potentially see gradual or abrupt, and are there any coordinated responses among these three groups? So the gradient we used is a mountain range in central Spain, Sierra de Gredos, and we mapped the bioclimatic zones on this gradient, and you see we collected lichens here until from the bottom into the alpine zone until 2,100 meters in this case. And we used two taxa that replace each other as you climb up the mountain. So Umbilicaria pustulata is in the lower level populations, and Hispanica is in the upper populations. And then in the middle of this gradient, there is this zone where both taxa co-occur. And these were ideal species for us because they occur in large populations. So we could collect 100 individuals here per population, and they were neatly spaced out 200 elevational meters. We collected the 100 individuals and sequenced them as a pool, this pool-seq approach. And then we had a sample of metagenomic DNA from this sequencing pool. And this metagenomic sequencing pool was then mapped against the fungal reference genome so that basically we obtained a genomic cross-section of the entire fungal population. And then we could do pairwise comparisons between these populations to assess differences. And we found that in both species, we see quite a clear distribution or a clear clustering of populations. And these correspond to a high elevation and a low elevation group, and a high elevation and a low elevation group. And the differences in the high elevation and the two elevation clusters were spread across the entire genomes. We found them in genes as well as in non-coding regions. And but this particular pattern was really strong. And if we map this on the mountain range, you see that the break within Umbilicaria pustulata happens right here between these two populations. And in Hispanica, it happens right here between these two populations. We also looked at the photobiont communities, and this was a little bit of a different approach. Here we used ITS metabarcoding. We selected 20 individuals per population and metabarcoded these samples. Altogether, there were 5 different Trabuxia lineages, and they were non-randomly distributed along this gradient. So OTU3 was at the bottom, OTU2 was only found in the middle, and OTU4 and 5 were only found at the top. So a very strong structuring of these OTUs. However, we also saw that OTU number 1, which is the lightest color here, was found in every single population, not in the same abundances, but basically everywhere. So we termed this one the climate generalist in this particular gradient. And I will now take you on an excursion to North America just for a second to show you that this pattern of altitudinal structuring is not uncommon in lichen photobionts. This is a different species now, and these are different gradients. I collected here 5 different gradients in California, and I found 4 dominant algal haplotypes, different Trabuxia species, and they show exactly the same behavior. So they have very clear altitudinal preferences, and they replace each other as we go up the gradient. So this niche partitioning along elevation seems to be a common pattern. We come back to the European gradient and we look at bacterial communities associated with these lichens. These were assessed with 16S metabarcoding of these samples. And we see in different measures here that alpha diversity is going down as we climb up the mountain. And this is seen in the individual species, the Pustulata and the Hispanica, as well as in the combined data. And that's a very commonly observed pattern that we also know from macroorganisms such as vascular plants or animals along elevation gradients. When we look at community structure within these bacterial communities, we see that there are 3 altitudinal classes. And when we compare, so these are the low elevation, mid elevation, and high elevation. And when we compare this with the climatic conditions, then we see that we're here going from cool and wet conditions to hot and dry conditions. So there's also climatic structure in the bacterial communities. And if we look at this middle part of the gradient where both host taxa co-occur, we can assess the influence of the host on bacterial communities. And here we see that the bacterial communities also structure according to host, whether it is Hispanica or Pustulata. So we conclude that environmental conditions as well as fungal host structure bacterial communities that are associated with lichens. If we put together all these three data sets, the genomic differences in the host and bacterial community turnover and photobiont turnover, then we can see that there are particular elevations where a lot of things happen. And I summarize them in this next slide here. So in this elevation between 800 and 1,200 meters, we see a genomic break within Pustulata. We see the lower distribution limit of Hispanica. We see the upper distribution limit of the alga OTU1, and we see the bacterial turnover. And in this higher elevation zone over here, we see the genomic break within Hispanica. We see the absolute distribution limit of Pustulata. We see the upper limit of algal OTU2, and we see a bacterial turnover. So we can speculate now that these locations on this gradient are ecological boundaries that cannot be crossed by certain genotypes of algae and fungi and bacteria. And they happen to be at a mean annual temperature of 11 to 13 and 7 to 9 degrees centigrade. So we could now compare this to other gradients as well. So we saw that changes along the gradient can be abrupt, they're not linear. Algal symbionts apparently show signals of spatial niche partitioning along an elevation gradient. And we see that the lichen holobiont responds as a unit. So all compartments, the fungal, the algal, and the bacterial components do respond, and they cause a particular setup or a particular combination in a given climate zone. So we wanted to know what happens at the physiological level, and we compared low elevation and high elevation thalli. And we found in this particular gradient here that photosynthetic, so we checked photosynthetic performance against the availability of water. So basically we let the thalli dry out slowly and checked how well they performed photosynthetically. And we can see here that the Mediterranean species, so from low elevations, they reach their photosynthetic optimum at a much lower water content of the thallus than the high elevation specimens. So there seems to be some adaptation to the environment. However, we don't really know if this is due to the alga or due to the fungus in this in the system, because we know that the fungal partner, the mycobiont, has a different genomic composition in high elevations than in low elevations. And the photobiont is also exchanged between the low elevation and the high elevation. So we try to see if we can get closer at this question, who's contributing really now, the photobiont or the mycobiont? So here we ask the question, what is the effect of the algal lineage on photosynthetic performance? And we selected thalli that had the same fungal partner. So they were both Pustulata with the high elevation genotype, but they were carrying different lineages of Streptococcus photobiont. And here we studied photosynthesis kinetics. So we dark adapted the lichen thalli, we gave them a light flash, and then we measured chlorophyll fluorescence. And this can give you information about how the different parts of the photosynthetic apparatus function and how effective photosynthetic performance is. And we saw significant differences here between the two thalli, which were carrying the different algal strains. So clearly, algal lineage has an effect on photosynthetic performance. But how about the fungus? We were also able to look at thalli that had different fungal partners. This is Hispanica and Pustulata, but which were carrying the same algal photobiont. Here they are carrying ODU1, and we see also here strong differences. In this case, two different microbionts are carrying another ODU2, other photobiont, again, strong differences. And in the last example, we had two different genetic lineages of the same fungus, and again, also strong differences. So we also see that fungal species identity and even fungal ecotype, genotype, do affect photosynthetic performance. Next, we looked at the transcriptomic response of lichen thalli, which had different fungal-algal combinations. And you see in this gradient that we selected here, low elevation populations have one microbiont genotype, and they always pair up with one photobiont lineage. And the high elevation individuals have the high elevation genotype of the microbiont, and they can carry two different lineages of photobiont. And here again, we gave them a light treatment, and we looked at the photobiont transcript, and we see that they very clearly separate by photobiont lineage. So that was a much stronger signal than the treatment. And then we also looked at microbiont transcript, and here you might expect that they separate by microbiont genotype, but that's not what we saw. Also, the microbiont transcript very clearly separate along the algal identity. So algal identity is really important in structuring the algal as well as the fungal transcriptomic response. So we see that algal strain is very important in influencing photosynthetic performance and even fungal transcription. And fungal genetic background also affects photosynthetic performance. So now I'll very briefly summarize what we found at the genomic level so far. The strongest signal was this division into altitudinal, a low elevation and a high elevation altitudinal group. We saw this in 3 species now where we could get population genomic data. And in each of these species, we find that these population groups group according to biome, basically, cold temperate versus alpine, or Mediterranean versus cold temperate. And this is happening also on different continents. The response is polygenic, so we obviously find hundreds of genes that are being affected. When we compare, especially these 2 groups are nicely comparable because they span the same climatic niches. We see that they are not doing it in the same way. So different sites and different genes are being affected. Some of these species are sexually reproducing, Hispanica and Fea. Pustulata is asexual, and we observe that they have different rates of change. So the sexual ones have higher rates of change along the gradients. So we conclude here that we see the same pattern of overall genomic differentiation, but each species is finding a slightly different route to climatic niche specialization. We looked at some of the functional genes that are highly differentiated along the gradients. And one group that we were interested in is biosynthetic genes. So these are genes that are involved in secondary metabolite biosynthesis. And we saw that some of these secondary metabolite biosynthesis gene clusters are entirely missing from either high or low elevations, suggesting that populations from low elevations can produce different compounds than those of high elevations. However, we could not measure them yet. This is genomic evidence only. And similar breaks we found in transposable elements, circadian clock genes, and temperature-associated genes. So all gene groups that are known to be playing a role in environmental adaptation. A quick look at genomic differentiation among Tribouxia. So this genus Tribouxia very much reminds me of Symbiodinium in corals. It also has only these very poor clade names, ACSI, that you have already encountered in Todd's talk. We don't understand very well how species are delimited, and only a handful of species is formally described. And molecular diversity is apparently quite large. So we were able to sequence 3 lineages of Tribouxia from the A clade and 3 lineages from the S clade. So these were found in Umbilicaria fair, the North American taxon, and they have warm and cold preferring distributions, and from Umbilicaria postulata from Europe. And here we also have cold and warm preferring distributions, and we have this generalist taxon that we find in every single population that we have analyzed. So we generated PacBio genomes and annotated them with RNA, and we find that these genomes were relatively contiguous and complete, and genome sizes varied a little bit, and number of genes also varied a little bit, maybe with the exception of this very first group here. And this happens to be the climate generalist. So this taxon with this huge ecological amplitude. So it is thinkable that this large number of genes is necessary to allow this taxon to occur in this diversity of niches. We compared genomic divergence within Tribouxia with these lineages that we had at hand here. And we see that there is a lot of difference between the different lineages. So the larger, so this is a pairwise comparison between all of these taxa, and the larger and darker the circle, the more similar they are. And we see that there are within, this is clade S and this is clade A. So within clades, there are more similarities, but even within clades, there is really a huge variation going on, as you can see in this example down here. So now we can look at evolutionary mechanisms that happen along the branches that lead to these taxa. And we looked at gene family, expansion, and contraction. And we found that overall, the signal of gene gain is really much higher than the signal of gene loss within the genus Tribouxia, which may also hint at the need for these taxa to have a lot of molecular functions that allow them to occur in these diverse niches. Then we also looked at GO terms of those gene families that underwent these significant evolutionary changes, and we found that things like developmental processes, transport regulation, metabolic processes, and signaling or response to stimulus were GO terms that, or were gene families that were affected, and these are quite strong candidates now to look into to understand environmental adaptation here. So in summary, I showed you that all parts of the lichen holobiont respond to the gradient, fungi, algae, and bacteria, so that the lichen holobiont in different environments consists of different combinations. we saw that there are physiological responses of both the fungus and the alga, and these responses may be dependent on the specific combinations of alga and fungus that we see in the thallus. And we also saw that genomics, population genomics, and comparative genomics approaches are quite useful in trying to understand what the molecular mechanisms are that allow environmental niche specialization. And with this, I would like to thank you very much for your attention and all my present and past lab members who have contributed to this research. Thank you.
So now we have time for a nice long discussion. So please, questions.
Thank you. I'm excuse me, I'm not really familiar with lichens, and I'm wondering whether the controlled common garden approach can be used with lichens for a complex G/E study like this, or if it just isn't feasible.
So it's very difficult to transplant lichens. People have tried it before, but with, I should say, with mixed success. So not every species is capable of doing this. So by removing such a specimen from the rock, you already mess quite a bit with its physiology. So maybe you could transplant entire rocks, but then they tend to grow only on the big ones, so it's tough. And one study that I know of where people have transplanted lichens that grow relatively loosely on soil, it took ages for them to see changes. And then they were also very mixed changes that were difficult to interpret. So it's definitely not as straightforward as it is with vascular plants, unfortunately.
Hi. So you, for the European species, you showed results for a single elevational gradient, or was that multiple elevation?
Yeah, so I didn't specify. So these are multiple studies really that I talked about here. I showed you the study on this long elevation gradient. That was a single one, but then we repeated for Pustulata. We could collect additional ones also outside of Spain, in Italy, for example, 1,000 kilometers away, and we found exactly the same division.
Okay, and so I was curious about how, in terms of population structure, is the fungus more strongly structured by elevation or by space?
By elevation.
Okay,
it's
so
completely structured.
So high elevation genotypes are more closely related to each other.
Absolutely, yes.
Than the low. Okay.
And in the Californian one, exactly the same.
And then I guess you could also do like a latitudinal comparison of the same kind, right? Because is that something that?
Yes, we haven't done it yet, but the specimens are around. Yes, we could do it, but it's quite difficult to collect this large amount of lichens in any one population. So for these particular, we looked very long until we found these elevation gradients where we could actually do such a controlled sampling. And if you go along latitude, it's not that easy to find such a beautiful sampling. So it was more fruitful for us for now to look at elevation only.
Is there any evidence that the elevational genotypes might be reproductively isolated, or?
We looked for gene flow, and we did find evidence of very limited gene flow among the high and the low elevation populations.
Hi, very fascinating. And I'm not at all familiar with the topic of lichens. I was just wondering, when you showed this altitude distribution of the algae lineages, the 4 and 5 seem to be kind of on the top and rather in the middle. And then in between, you found a lot of the, I think it was a generalist number 1. So is the reason for that the number 1 is generally so dominant, or is there something more than altitude that defines this distribution?
The generalist, according to presence absence, is found in every single population, but the abundance is much higher in the higher altitude populations. And with regular Sanger sequencing and barcoding, you would never detect it in the lower population. So it needs to be a meta-barcoding approach to even detect it. So it is able to be there, but it is not, maybe it's there in low abundance, or it's not preferentially captured by the lichens in the low elevations. So we don't know this. So we just know it's theoretically possible that it is there. And the high elevation genotypes, they, in this particular gradient, they were, we found them only with the 1 taxon, but we know them from our latitudinal whole range sampling of the other taxon that they also occur in that one in the very cold areas. For example, populations in Scotland all carry this cold genotype of Tribouxia. So similar signals here. But what was your first question?
I was wondering because 4 and 5, you know,
Yeah,
they kind of showed up at the very top that you found them. And then you showed them again very dominant in the middle altitude, there where they start kind in the distribution, but not in between. In between the 3 samplings had a lot of the number 1, I think it was, right? And so I was wondering whether this was just incidentally because maybe of low numbers of samples.
Yeah, maybe we didn't find it. Yeah. So modeling showed that these are the cold adapted genotypes, and we sampled to analyze these taxa only 20 individuals per population. Maybe it just wasn't sufficient.
Thank you. Hi, I was wondering here, how well is the role of the bacteria understood that you find in the holobiont, and are you looking at all in your genomic and transcriptomic data into the transcriptomic changes you may observe in the bacteria, and can that help you to understand their role?
We haven't done this yet, so we haven't looked into the portion of bacterial genes in these gradients. The role of bacteria on lichens are not so well understood, I would claim. Even the specificity is not really well understood. The communities are highly complex. Some people claim that there are some bacterial lineages that have nitrogen fixation properties, and maybe that is a useful feature for the lichens that occur in very nitrogen poor environments very often. But at this point, we don't know very well what the bacterial communities do. Only we see these extremely strong structures. But for now, we have concentrated on the main partners first, because even here, we are still quite at the beginning, right? We are here producing de novo genomes, so the bacteria might be a second step then.
Yeah, I have a question in particular to those genomes. So when we compare seed plant genomes, what we see is that the additional genes or the missing genes are frequently genes of unknown function, or maybe as annotated in specialized metabolism. So brutally said, they're boring in the sense that they're not interpretable. And we see most of our changes actually in cis, so gene expression changes and presumably promoter changes. Do you see the same, or do you also have genes among your presence absence variation that are kind of cool because they're interpretable?
We don't know yet, so this is very fresh data. But of course, the number of genes that are unknown protein that are connected to unknown proteins is really high, In vascular plants, it's already high, but in these algae, of course, it's even higher. We don't have a lot of genomes in that are closely related to these Treubuxia lineages. So it's a problem that we very often don't know the functions of those genes where we see very strong differentiation, and gene expression we haven't looked at yet.
Hi, you mentioned that you saw some differences along the elevational gradient in transposon content, or was it transposon content or identity? And like, do you see any interesting, like, do they have effects on genome rearrangements or anything on the structure of the genome?
We were hoping that. So this was also a really explorative study because it was the first one where somebody looked at transposable elements in a lichen genome. And it took us forever to find our way through these data because you couldn't be sure it's really a transposon and so on. So here we looked at diversity and insertion sites of these transposable elements. And then we were hoping to see whether they are adjacent to any genes that might be of interest. But we couldn't really see a very strong signal of something. So where we had said, okay, here is a trehalose, that's a sugar that's known to enhance frost resistance or so. Unfortunately, we didn't see this, but we saw a strong differentiation again in these high elevation and low elevation groups, also based on transposable element insertion sites, for example.
Very interesting. So going back to your initial this diagram that the biotic response is nonlinear. So now thinking about your three zones, the high, the low, and the middle. So which part is the linear? Which part is the nonlinear?
Okay,
and what causes that?
So the transitions correspond pretty well to the transitions of the major biomes that are assigned according to vascular plant biology, so or vascular plant community composition. And so the transition zone, the steep transition, is between the Mediterranean and the cold temperate, which was here at
the middle zone.
The middle zone is the cold temperate, and so that would be a stable part of this. And then we have a transition again, and then we had the alpine.
Okay, and so the transition zone is there a gradient that can, so the from the lower to the higher, so there's a great gradual change, or it's totally different community?
In the algae do more or less a community turnover. The fungi have a completely sharp genomic break. So we know in which 200 meters of elevation this is happening. So we can specifically target this area, and we know everything collected below is going to be this fungal genome. Everything collected above is a completely different fungal genome.
Wow, amazing. Thank you.
Further questions? Rashid?
Just a quick question. Is there evidence of reticular evolution amongst these fungi? So when you talk about areas which you have, related species living in close proximity, you have regions where they are more isolated from each other. When you look at the genome data, do you have any evidence of integration or any other such things happening between these two?
We haven't looked at it.
Okay. That's something that's they remind me of land corals, and I was thinking this is very common, cryptic speciation, where you have this, and I was just thinking it might be an interesting way to look at, because you have two very close related species here, right? So that is kind of an interesting way, and you have the possibility for sexual reproduction. I guess they have apothecia and so on. Yeah,
one of them is sexual, the other one is asexual.
Oh, it is? Okay. might be something to think about whether there might be evidence of the genomes being intermixed in the transition zone, creating novel lineages that are maybe have more different alleles and combinations that may allow them to be more flexible. Just an idea. If the selection, because it sounds to me like the biodiversity here kind of peaks in the middle. Is that true, or?
What do you mean by biodiversity?
I was just thinking, if you had to protect lichens in the future, which part of this distribution would you think is the most highly diverse genotypically? And
oh, I think we can't tell by this one gradient.
Okay.
I mean, from the bacterial community, it was the same pattern that we observe along gradients.
Okay.
That has been known for a long time, that diversity declines as we go up. Then, of course, there are also some studies on some organisms that claim that the middle zones have the highest diversity. But from the data I collected here, I wouldn't be able to say yet where the holobiont diversity is greatest, really.
All right, thank you.
Okay. There are no further questions, then please let's join hands and not join hands.
Thank you very
clap
much.
hands.